Hello, everyone. Thank you for joining us today for this installment of Boyce Thompson Institute's Breaking Ground discussion series. My name is AJ Bushy, and I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome Jim Giovannoni, a plant biologist here at BTI, whose lab is focused on the molecular and genetic analysis of fruit ripening with an emphasis on tomatoes. And today we'll be talking about the relationship between fruit ripening, nutrition, and consumer quality. Hey, Jim.
Hey, AJ. Hello, everybody.
Thanks again for joining us today. And let's kick things off. Could you tell us a little bit about yourself and how you came to BTI and doing the research that you're doing today?
Sure. I'm originally from California, so I'm still been here 22 years, but I still love it when it snows out. I grew up in the San Francisco Bay Area, went to undergrad at UC Davis, got my PhD at UC Berkeley, so I stayed in the Bay Area. Did a postdoc out here at Cornell with Stephen Tanksley, and then took a faculty position at Texas A&M University. Had 8 great years in Texas, and then moved back when an opportunity came up, particularly working with the USDA ARS, but housed here in BTI.
So you are with the USDA technically, that's right?
Correct. Yes. So I'm a USDA employee. Thank you all. I'm a taxpayer supported employee of the U.S. government. There's a USDA building directly across the street, and I'm actually the director of that. But my lab has always sat here in the Boyce Thompson Institute, which is a great opportunity to take advantage of all the expertise and all the infrastructure and all the great people that are here at BTI.
Well, we're glad to have you. We're glad you're here. You want to tell us a bit about some of these folks in these pictures?
Sure. So this is the current lab. The selfie there with the top of my head in the way is out in our annual planting of our field in Freeville, New York in early June. And then the 3 folks on the left weren't able to join us that day. But that's the current lab, so about 10 people, looks like, and a combination of technicians, postdocs, research associates, and PhD students.
And you study primarily tomatoes. Is that your like number one fruit, or is that the only plant you study?
So certainly number one. So we the lab's especially interested in fruit biology, fruit development, and particularly fruit ripening, that terminal stage of fruit development where the fruit makes this incredible transition from being something very firm, green, not very appetizing, indeed harboring some chemistry that might make you sick, to something very attractive, both visually, in terms of texture, in terms of flavor. And we're really interested in how that transition occurs. We do work a little bit on some other crops. We've got a student right now who's working on melons. And we often work with collaborators on other species who look at some of the genes we've identified in tomato and ask if they're doing similar things, for example, in an apple and a strawberry and a peach and so on. But we do most of our work in tomato. For a number of reasons, it's a really tractable system. So you can imagine you put a seed in the ground and you have a ripe fruit in about 3 months. That makes doing experimental science much easier than, say, if you're working with an apple tree where you might have to wait a couple of years just to get decent fruit set. Also, it's a pretty valuable crop in terms of farm gate value. It's about $3 billion a year in the US alone. Tomatoes highly consumed. We consume about 70 pounds per capita in this country. About half of that is fresh and about half in processed form. And that's really things like ketchup, pizza sauce, tomato sauce, and things like that that go into a lot of the foods people really enjoy. It's also a really good source of a number of important nutritional compounds, particularly vitamin A and vitamin C. I think a lot of people think of vitamin C, they think of citrus, and it's certainly an excellent source, but given our large consumption of tomato is actually the primary source of vitamin C for those in the US. And beyond some of the specific vitamins that are found in tomato, it's generally just very high in antioxidants. A lot of that is due to the carotenoids, which make it that nice bright red color you see in the picture on the screen right now.
So it sounds like there are so many grown, they must be mass produced. And so how does this mass production, work to get to us? How can they make it? Because when I think of tomatoes, like when I grow tomatoes, they lately they've just been splitting open, but they seem to rot pretty quickly. So how are they able to do that in the industry?
Yeah, I mean, so much of the tomato that you'll find in the supermarket is grown, in very high production areas. So in the US, we grow a lot of fresh market tomatoes in Florida, a lot of processing tomatoes in California. And also there's a lot of fruit grown in the upper Midwest, in places like Ohio, along the coasts, down in North and South Carolina. And these fruit are, as you can imagine, optimal seasons are going to vary in all those different places. And so that means, like many is the case for many fresh fruits and vegetables, we have high intensity production areas. And then things need to be shipped. And at different times of the year, they're being shipped from different places. And in fact, I think everybody who enjoys a tomato once in a while knows or at least if they go to the supermarket, they know they see them always, right? 12 months out of the year, there are tomatoes there. So that means they're being sometimes produced south of the border, Central South America. Often, actually, many will come in our area in particular from north. There's big greenhouse industry in Canada, so they'll be shipped in from that area at certain times of the year when there isn't outdoor production or Central American, South American production. And if you look carefully at the little stickers you see on your tomatoes and many other fruits, they'll always have an origin. And sometimes you'll even see tomatoes that are produced in Europe showing up in our supermarkets. So they're shipped very long distances often. And that's the case for many fruits, not just tomato. Banana here is a good example where virtually you'll have no local production unless you get some maybe down in South Texas or South Florida. But even there, it's primarily going to just be maybe you have a plant in your backyard. So these things are being produced and shipped all around the world. And so that becomes an issue. How do you get it stable for that long period of travel and then make it through the food delivery system and then to the consumer, where the consumer is also going to expect a little bit of shelf life. They don't expect their fruit to decay overnight. They anticipate a few days at least before they have to consume it, or that it'll be stable that long. So there's a lot of effort put into the fact that there's this long shipping time, and a lot of things become important. Texture is one of them in particular. So firmer fruit are much easier to ship than soft fruit. You can imagine if you pile fruit even a little bit deep and they're too soft, they just squish each other. So firmness is very important. And ultimately, the way that this is dealt with, it's in a number of ways, but it all comes down to the same thing. It comes down to delaying ripening. So that's done either by just harvesting early, so you have time until the fruit fully ripens before it reaches the consumer. And during much of that time, it's still firm, it's still unripe. We use genetics. Tomato is a good example. There's one gene in particular that's bred into many fresh market tomatoes that delays ripening. So it's a gene that's involved in the regulation of ripening. If you knock it out completely, you have two copies. It's a tomatoes, what's referred to as a diploid. We have two copies of every gene, two copies of each chromosome, one from each parent. If you have both of those genes defective in this particular gene called RIN, a ripening inhibitor, you get no ripening. And that's actually what that tomato up on the left is. It's a RIN mutant. If you have one copy, so you breed that mutant with a normal and you get a hybrid, which is what most of the tomatoes you see in the supermarket are, you end up with a slow ripening fruit. So you can use genetics to give you time. You can harvest early to give you more time, and you can hold fruit in a controlled atmosphere. So low temperature, low oxygen, high CO2, all of this will delay the ripening process. And that gives the production and delivery system time to get that to the consumer.
So how does that impact the flavor?
Yeah, so that's the problem. And Mike, if you could, there you go, thank you. The result ultimately of all of these methods is incomplete ripening. And just like eating any fruit that you pick a little too soon, you typically have, losses in terms of flavor, in terms of nutritional quality, in terms of that kind of right feel in your mouth that you expect when when a fruit's, fully ripe and maybe a little more juicy, and ultimately a lot of consumer dissatisfaction. And tomatoes really, I think, something where consumers realize that maybe more than other fruits, because I think most people at some point have probably grown a tomato plant. I always say, even if you're living in a big city, if you've got something on your balcony or on your windowsill or outside on your front step that you eat, it's probably a tomato plant that you've got in a little pot. So people know what they should taste like. And of course, those homegrown varieties, the ones that are marketed for home use, never carry the kind of mutation that I referred to that'll delay ripening and give you that firm but not fully ripe fruit. So I think tomato in particular, people know what they should taste like, so they often are extra disappointed with what they get sometimes in the supermarket. And that's not to say breeders have done a great job. The fact is, there's a constraint. That constraint is to make, people see when you go to the supermarket, fruits and vegetables are expensive. And in efforts to try and keep that cost down, breeders really have to come up with varieties that'll, produce well, that will have disease resistance, that will manage that long-term shipping situation, and that will still have acceptable quality. And the fact is, breeders have done a great job in developing a lot of varieties that are pretty good. But I think still consumers often feel that they could be better. And I think sometimes we notice too that fruits in particular, the quality doesn't seem what it was a number of years ago. And some of that's just we get older and we remember the good things and ignore the bad things. But there's more and more large scale production that is then feeding into delivery systems that ship things further and require more of that shelf life to get them there. So you have more and more fruit on the market that was probably picked early, that was probably held in low temperature, some kind of controlled atmosphere before it ultimately gets to the consumer. The trade-off for that is, I think, also those of us who are a little older and think back, we see more and more fruit in the supermarket all year round that maybe we didn't before. Just think about something like peaches and plums. Now you can get those in the middle of winter because they're coming from South America. Yeah. So we trade that kind of desire and probably consumer demand for access year round for a loss of quality.
Yeah, I think that's a good point. Instead of, eating things when they're normally harvested, we have them all year round. And
right.
A little spoiled in that respect.
Yeah. And I always say, if you want the best quality, pick what's in season at that time. And that's, that's most likely to come from a more local supplier. It's most likely to have undergone less control to keep it firm and have proper shelf life. And also, it just hasn't, whatever it is, it probably hasn't been in storage for as long.
Right. It might actually be ripe. Might actually have been picked when it was ripe and not underripe. So you mentioned this gene, the ripening inhibitor gene. What else do we know about the genetic underpinnings of the ripening process?
Well, quite a bit. I think certainly we know that the hormone ethylene is important for ripening of many, many fruits. If you block ethylene, you delay ripening. If you supply ethylene to an unripe fruit, you can promote ripening. So we know ethylene is very important. We've known that for a long time. Ethylene control has been a means of controlling ripening that's been used for decades. As I mentioned, there are some mutants, and this slide that's up right now is just a picture of a number of interesting tomato mutants that have different characteristics. And a lot of what our lab does is we study these mutants because ultimately they represent genes that are defective. And because of the fruit don't ripen normally, we know these are genes that are important for the ripening process. So we've spent a lot of time trying to identify genes that underlie these different mutations. These are all natural mutations. So these are basically things that have been found in some cases in greenhouses by tomato geneticists or producers, or in some cases out in the field. And as you can see, most of them don't look quite normal. Some are very clear, like the ones up on the top. Several of those have what look to be very incomplete ripening, particularly the ripening inhibitor and the non-ripening. And those orange colored terms just refer to, now that we know what these genes are, they fall into a number of categories. Some are transcription factors. These are kind of regulators of other genes. The four up on top are all transcription factors. The ones in the middle that are referred to as signal transduction components, a lot of these have to do with ethylene. So for example, the never ripe mutation, the second kind of orange colored one, this is a mutation in an ethylene receptor. The non-ripening ethylene insensitive down on the end is a mutation in a gene between that ethylene receptor and the regulators that ultimately promote the genes responding to ethylene. High pigment, the one right in the middle, is a little different. That's a gene involved in light perception. And you can see that one actually looks kind of bright red.
That one looks really good, yeah.
Yeah, and so that's basically, that mutant thinks it's seeing more light than it is. And the result of that is it produces more carotenoids to protect itself from high light. So carotenoids will help absorb some of that light. Some are involved in photosynthesis. So they're actually involved in that conversion of light energy to chemical energy, but they're also used as protective compounds. So this particular mutant will produce a lot of carotenoids because of that essentially misperception of how much light there is in the environment.
So even though it looks ripe, it would still taste bad. It would taste like
no, this one actually tastes pretty good. It has other problems though. That mutation isn't specific to the fruit, so it results in plants that are kind of dwarfed. They don't produce as much fruit. They're also brittle. So it's actually a mutation that would be very desirable in a fruit specific form. And people have actually shown that you can do that with GMOs, with transgenes, but those haven't hit the market because because of all the issues that the difficulties in dealing with GMO products. But the mutant itself has some other issues that just make it hard to work with. Breeders have certainly used it. It is in some varieties. And then the last set down there are involved in plastid development. So you can imagine the plastids are both what make or hold the photosynthetic machinery and make green tissues green. And a tissue like a ripe tomato, those plastids become what are called chromoplasts, and they store a lot of carotenoids, which are what is making the fruit red in this case. So you alter that and you get some changes in color. So, and a lot of these, or at least several of these, are actually used by breeders. So one I mentioned, ripening inhibitor, up on the top corner. If you have 2 copies of that gene mutated, the fruit look like those on the left. They just don't ripen very well. But if you have a heterozygote or a hybrid, you get a long shelf life, slow ripening fruit. And probably a lot of people recognize that kind of coloration, a little bit lighter, maybe not fully ripe looking, that you see in a lot of the fruit you get. In the
very familiar.
Yep, a lot of, particularly the larger fruit. The mutation is not used so much in the smaller cherry varieties, but certainly in larger fruit it is. And then another one that's widely used is uniform ripening. And this is found in almost all more modern tomatoes. So, and those of you who might be in the audience who grow heirlooms, are interested in heirloom tomatoes, or just look for them in the farmer's market or the supermarket, you'll notice that heirlooms will typically have that kind of discoloration on the top. They're not uniformly ripe. And that's really the wild type or normal tomato. This mutation gives you uniform color. So that gives you a fruit where you don't have that greenish or sometimes yellow or whitish coloration on the top. And it's bred into most commercial varieties because in general, the feeling is the consumers like that uniform color. But seeing that incomplete or not uniform color in a variety is a good sign that you're looking at an heirloom. So those will often be a hallmark that someone interested in heirlooms will look for. But that's certainly a prevalent mutation that's bred into a lot of tomatoes. Another, again, that I just mentioned, high pigment, is not so widely used, but it does give that very deep coloration and dark red flesh. And the last one is green flesh. Some of you may have purchased Kumato tomatoes, or these kind of almost brownish colored, kind of darker green on the top. Depending on the variety, they can look almost brown. In fact, the same mutation in pepper are what are called chocolate tomatoes. They are much more brown. So that's a mutation that results in plastids that don't fully convert to those just containing the red carotenoids. You still keep some of the green chloroplasts. And then if you remember being a kid, taking a green and a red crayon and scribbling them together, you get kind of a brown color. That's what a Kumato tomato is. It carries that mutation. And with that, they have a little more of a vegetative taste that a lot of people like. So certainly one worth trying sometime if you have it. But so those are just a number of these mutations that we've looked at, trying to better understand how are these genes that when they're disturbed or broken or mutated, they have these pretty significant effects on ripening. So what are they doing in their normal state to control that process? That's something we've been interested in our lab.
So what have you found in your studies as you research these genes?
Well, a lot. So we know that there are, quite a few transcription factors. We probably focus there the most. So these regulators of other genes. We know a little bit more about how they interact with each other. We know about some of the ones beyond this collection that I just showed in the last slide. This one that's shown here is an interesting one. It's called LOB1. The name up there is SLOB1, which just sounds kind of fun, but that's because Solanum lycopersicum, which is tomato, and then LOB1 is the name of the gene. LOB stands for lateral organ boundaries, which is a name of another gene originally identified in Arabidopsis that is involved in how organs are defined, their boundaries are defined, and the tissues separate. So this similar gene, the name is just because it has a similar DNA sequence to some of those previously described genes. This was a gene we identified looking more specifically at the inside part of the fruit, the locule, the gooey jelly stuff that surrounds the seeds. And we were interested in that because early in fruit development, that's very firm. And then it goes to that kind of liquid state. So we knew a lot that was controlling texture was there. So we were interested in trying to identify transcription factors that might be a little more specific to texture. Most that we had looked at previously control all of ripening. That is, the fruit don't change color, or they're altered in their ability to change color. They're altered in their texture. They often don't produce as much ethylene. The sugars and acids related to flavor, in addition to the volatiles that are related to flavor, aren't as produced as much. So kind of everything that you think of as ripening is impacted by a mutation like RIN. But LOB1 was not a natural mutation. That was a gene that we saw was highly expressed in that locular tissue around the seed. So we made our own mutant. We made a transgenic mutant and asked what happens. And it was interesting because if you look at the two sets of fruit from lob number 3 and lob number 6, those are just two different plants where we mutated this gene. And you can see they actually look pretty normal, except they don't decay as fast. And a measure of that is how much water they lose. So the two colored lines are the mutants. They don't lose much water. The black line is the wild type. It's called AC up here. That's just the variety name. So that loses a lot more water as they mature. And you can even see they start to rot, collapse much more than the lob mutant. So this particular gene, turns out, primarily regulates changes in the cell walls of the fruit and how the fruit soften. And so they're also much firmer than the wild type fruit. So this gets back to that kind of notion. In an attempt to control firmness, we end up, having all of these unintentional or undesired consequences on the fruit as they mature. So this looks like possibly a gene that could be targeted to address texture more specifically. So it's a new one we've just characterized in the last 2 years or so. So we're very excited about this one.
Right. So it sounds like theoretically you could it could ripen and have all the sugars and have nice color and nice flavor, but not get all soft and mushy to increase the shelf life. So you could wait and pick it when it's ripe and then ship it across the country. Is that
yeah, that's the idea. That was the original idea of those familiar with Flavor Saver, the first transgenic plant product, right? That was an attempt to target a gene that was thought, for a lot of good reasons, to be the primary regulator of softening. Turned out it wasn't by itself able to do virtually anything in terms of softening of the fruit, very, very little. But that was the same notion, right? How can you specifically target this one aspect of the ripening process that's a problem without having adverse effects on the quality of the fruit? And so, yeah, that's we're hopeful, and we're, some of the things we're doing now, we're starting to look at kind of diversity collections of tomato to ask how is a particular gene being expressed in everything from the wild progenitors of tomato through the first domesticates through heirloom types up to modern varieties. And can we find variation that then might be useful for breeders? Now that we know about a gene like this, could they specifically target lines that maybe have less expression and bring about a firmer phenotype?
That's a great idea. That sounds good to me.
We think so.
So what things are you looking at in the future? What are your next steps? You mentioned a few there. You got anything else?
Yeah. Oh, yeah. There's always plenty else. So, at the end of the day, what really motivates us is an effort to try and understand, the regulation of ripening as a whole. So we use things like mutants because they give us insights. I always say, if you never really, unless you have a lot of interest, you don't really understand how your car works till it breaks down, right? Something breaks, then you figure out what broke, and now you've learned something about how that system operates. So that's what we do with mutants. So we're trying to identify what are the other genes that are involved in control of ripening? We do that in a number of ways. We use genetics, we do crosses and we try to map regions of the genome that influence the ripening characteristics. We just look for gene expression that tracks with ripening. What are the genes that come up when ripening begins or goes down when ripening begins? That's a good source of candidates. We also look more specifically at the fruit. If you've ever looked at a tomato on the vine in your garden, it doesn't just go green 1 second, red the next second. They tend to kind of ripen from the bottom to the top. There's a progression. And if you cut the fruit open, you'll see that progression in internal tissues as well. So we've looked at those kinds of gradients of change of development to ask, can we find out what's changing in one tissue that's different from an adjacent tissue that's not ripening? And then finally, as I mentioned before, we're looking at genetic diversity. With the advent of relatively easy genome sequencing, we can now capture the genetic diversity of many tomato varieties and ask, how are the genomes different? How is the gene expression different? And how does that translate to phenotypes we're interested in, particularly ripening? And can we use that to understand not just where are the sources of variation breeders might use for practical product development, but also what can we learn about that variation that might tell us how genes are expressed, for example, or how genes might be influencing together in systems, in complexes, regulation of genes. So we've spent a lot of time on what are the key genes. Now we're trying to ask, how do they actually work? And then how well conserved are they when we get out a tomato? How much of what we find is a tomato thing? And how much can you translate to peaches, apples, bananas, anything you might be interested in? And then finally, very quickly, because I'm sure my student Matthew's on and listening, we do look at other systems as well. In this case, we're asking a very interesting question looking at melons. Because in melons, you have varieties that require ethylene to ripen and others that don't seem to care so much about ethylene. And we're trying to understand why that is. So maybe that'll tell us a little more about how ethylene controls ripening.
How much of these are conserved across families? Because when I tomatoes, I know are in the Solanaceae, and there's tons of crops, potatoes and eggplants, all kinds of stuff. Melons are cucurbits, right? So, I mean, is
Perfect.
What's that? Did I say it wrong?
No, you did. You just kind of gave this dismissive wave.
No, I didn't mean that. I didn't mean it like that. I'm curious because you've mentioned apples a few times.
Sure.
Do a lot of these fruit ripening genes, are they conserved across all these all the different angiosperm families, or for the most part? Or
yeah, so some certainly are. Some certainly are. And so a good example is that, probably the best studied is the ripening inhibitor gene. This is a member of what's called the Madsbach transcription factor family. The plant scientists will recognize that from genes that are described in terms of their functions of floral development, and the fruit's part of the flower. So that makes a lot of sense.
Okay.
But that's one where we've looked at many different fleshy fruit species and have even done some transgenics, particularly with collaborators. And so, for example, you can go to melon and identify the equivalent gene, knock it out, and create a non-ripening or a slow ripening melon. You can go to strawberry and do the same thing. You can even go to banana, which is a monocot. So there the evolutionary divergence is particularly large, and you still find similar genes doing similar things. So absolutely, and that's one of the things we're interested in kind of understanding. How well are these regulators conserved?
Okay, I think I'll throw out there, we can start taking questions now. If anybody has any questions, feel free to put them in the chat box. I'll just ask a quick one following up on that. Has there been work done on like gymnosperms, like pine cones? Are these in there? Do they affect do they go that far back evolutionarily, or?
Well, you can certainly find, the similar, the homologous gene. You certainly find homologous genes. It's hard, when you're looking at a system that doesn't have, or a plant that doesn't have a fleshy fruit, to know exactly what they're doing. I mean, the genes are largely conserved. Whether the equivalent gene that would have that function in tomato, which one it is in some of the non-fleshy fruited plants, that can all be determined. I'm not sure there's been a lot of effort to do that. And then to actually understand what they're doing, that would require work. It could certainly be done. It's not something we've done. But what you can do, you can get a sense, for example, looking at Arabidopsis, where you have a fruit, but a very different non-fleshy fruit structure. And so, for example, some of the similar genes to Rin are actually involved in how the silique, which is, more like a pod, how it opens, how it breaks. And so it's, you'd say, well, that seems very different than how the flesh, collapses. But actually that biochemically, there's a lot of similarity in what it's regulating. And functionally, it's actually very similar, right? Because whether you're busting open a pod to throw your seeds out, or whether you're making a fleshy fruit that becomes attractive to some animal to consume and then deposit the seed somewhere else, it's all about seed dispersal. So in some very cool ways, you have similar genes doing very different things, maybe not so different, but in the context of very different tissues to bring about the same kind of end.
Yeah, that brings up something I was thinking about. I tend to think of like ripening and rotting is all kind of the same thing, right? It's just at one point we decided it tastes good, so now it's ripe. But I think it's all kind of one big process. I don't know if that's the right word.
Yeah. I mean, well, so certainly it's a progression, right? And you typically will you'll have ripening, and then you'll see rotting, and it's again one of those things that, unless you really think about fruit, you'd never think of and you'd never care. But if you think about it, you'll notice you very rarely see an unripe fruit rot. It's always ripe fruit that rot. And the reason for that is a lot of the same reason they get soft. It's that textural breakdown of the cell walls and other constituents of the fruit, how much water, how much turgor the fruit has. As those things change, in general, you get, as you have that breakdown, you just get a general increased susceptibility to stuff that can get in, right? The barriers are weak. And now whatever microbes that otherwise might not have penetrated and infected the fruit start to. So then you get decay and you get rot. I'd say, biologically, they're very different. Ripening and rotting are very different processes, but they're certainly associated, right? Once you ripen, you often begin to rot.
So
become predisposed to rot.
And so,
and sorry,
that's all right.
That rotting is, that's probably escape plan B, right? So if the fruit is not consumed by something that releases the seeds, right, you don't want the seeds trapped in this very stable vessel that holds them forever. You want them to get out eventually. So
it rots on the ground, and
yeah, they get out and the microbes liberate the seeds rather than a bear.
So that's a good point, because if this is all to disperse the seeds, right? That's the whole point of fruit, really, right? To disperse the seeds. Are some of these signals that you're talking about before, are they coming from the seeds? Is it like once the seeds are ripe, the seeds are sending like chemical signals to the fruit to start ripening or softening or whatever the right term is?
Yeah, no, it's a really good question. It's something we're interested in. And the short answer is just think about many of the fruits that you consume, right? So we eat a lot of seedless citrus, seedless grapes. Now everybody likes seedless watermelon. So on the one hand, it doesn't seem like the seed are that necessary. That once you've made the decision to start developing or expanding that fleshy fruit, ripening seems to happen, whether the seed is there or not. So that's something we've been interested, because like you say, it makes a lot of sense. This is all about seed dispersal. The fruit exists to initially protect the developing seed and later to facilitate its release. And that can happen in many different ways. In some forms of fruit, you have something that, basically the fruit's providing protection, and then you shatter or break open and release seeds. And others, the fruit tissue itself might serve as a vehicle. If you've ever taken your dog for a walk and runs through the woods and comes back with burrs all over it, that's a mechanism of seed dispersal, where in that case the fruit is essentially got projections that help it hang on to the fur of animals and let them disperse that way. So there's a lot of mechanisms for getting seeds out there, but ultimately that's the ripening process. In terms of the fleshy fruit, it's to release the seeds through something consuming it. And so it does make a lot of sense that kind of development would be associated with the seed may be saying, I'm ready now, let me out. and I'm not sure that it's not the case, but certainly the fruit knows how to do it without the seed. That's pretty clear. And former PhD student in the lab down at Vantage did a lot of nice work showing how very similar fruit without seed is to a fruit with seed. There are some differences. One of the differences is in that gooey locule, which doesn't get so liquid, or at least so early as it does when there are seeds there. But in general, the fruit seems to be able to do okay without them. And that some of that might just be, you can imagine from an evolutionary perspective, you've got a cluster, say, of berries. If most of them have been pollinated, most are developing seed, you might have a motivation or a selection to ensure that all the fruit kind of ripen together, even the ones that never had fertilized seed.
Right. Makes sense. All right, I think we're going to try something we haven't done before. We got a couple of hands up. So we're going to allow some folks to talk. Are you up for that, Jim?
If you'll let them talk, I'll listen.
All right, David Silberman, we're going to go to you. Let's see. I need to ask you to unmute.
Okay, can you hear me okay?
Yes.
Okay, yeah, it seems that at least in my chat box, it was disabled, so I couldn't ask a question.
Oh, huh.
But that's okay. I orally is okay as well. So my question has to do with looking beyond the tomato plant for specific gene sequences that might reside in other plants, even other microbes that have a somewhat similar sequence, but not quite the same thing, that might affect some of the traits that you're looking for. And I know that a tomato is a berry, and I don't know if it just goes beyond looking at that particular family, but you never know where gene sequences are hiding that contain those sequences that might be of interest. And often we see one that's many base pairs, but then there's maybe a dozen that are a little bit different that might have an impact. So is there any looking beyond the tomato plant?
Yeah,
I won't mention microarray analysis.
I'm sorry, I didn't hear the last part.
That's okay. That was for AJ. Usually I ask a question about if anybody's using microarray analysis to look for genes, but there are other techniques as well.
Yeah, I mean, no, I appreciate the question. And we look at, now in the genome era, so we look at we try to look at whole genomes often, where we do capture not all, but most of the gene sequences. So we can just see what is in the genome that might be similar, for example, and could similar genes have some similar functions, or often interestingly, very similar genes might have very different functions. Some of that just has to do with where and when they're expressed. So, and even there are a couple of examples of some of these ripening genes that do have multiple functions. So there's one called TAGL1, where the same gene early in development is influencing the division of cells. So, and it influences how big the fruit actually get. And then later, it essentially shuts off and then comes back at the ripening process and influences how the fruit matures and ripens. And there's very little cell division going on at that stage. So there's a case where you have same gene doing two different things. But we do a lot of expression analysis. So when microarrays were kind of, when they initially came out, there was actually a nice system was set up here in BTI. A lot done by the current Vice President for Research, Paul Debbie. We were doing microarrays at that time. Now we do still do a lot of what's referred to as transcriptome profiling, but we do it using sequencing-based approaches, RNA-seq in particular. So, for example, when we look at something like at a maturing fruit, we find, of course, the RIN gene, but we find there are close to 100 members of that family in tomato. And there are quite a few who are expressed in the ripening fruit. And there are others that have been shown, if you knock them out or overexpress them, to also have ripening functions. And this is something we're actually really interested in now, is when you remove one, or maybe just looking at varieties where you have different levels of expression, are there other genes that are similar that might be playing a similar to identical role that maybe don't in a particular variety where one is very predominantly expressed? But when you look at a mutant now, can you reveal some of these other genes that might have similar functions? And then I think, going on to your broader part of your question, David, then we ask, how well are these things conserved in other species? And we do look at expression data for many different fruits, because lots of people have generated that kind of data. And the nice thing is it's easy to pull it up and look at it. You certainly see very similar types of regulators, at least being expressed in the maturing fruit. So I suspect they're probably doing similar things.
Thank you.
All right. Thank you, David. We have a couple questions in the chat box. Could the microbiome play a role in the ripening processes in fruits?
Yeah, so that's a good question. I think, in general, there's certainly much more appreciation for what microbiomes are doing and how they're influencing many aspects of plant development. In fact, we have, with a collaboration with Jenny No-Niffen down the street in the horticulture department, we've done some experiments looking at altered microbiomes, and specifically asked, the student was primarily interested in more kind of general productivity questions, how much biomass do you get, things like that. But we did do an experiment where we looked specifically in a system where we had some variation in microbiomes. And that was done by essentially taking a specific microbiome and then sterilizing it, using that as a control, but then still providing nutrients. So you didn't, you could live without the microbiome in that case, or at least the what you started with in the soil. And those experiments, at least, we didn't see a lot of differences in fruit maturation, fruit ripening, fruit quality types of traits. One, to the extent we looked at them, and two, within the context of our measurements. And our measurements were pretty straightforward and simple things like, color and accumulation of carotenoids that contribute to color, texture, time to ripening, so it didn't seem like we saw a lot of differences there. But I don't doubt for a second that the microbiome can influence. This was a very specific experiment looking at a very specific variation in microbiomes. But I suspect that the microbiome, I think as we're appreciating much more now, is contributing to many things.
And we have another one. Could you please talk more about the melon case? Recently, a couple of genes have been reported as ethylene/maturity related, but still results are not as clear as a tomato. What is your experience with it?
So yeah, it's a good question, and it's really why we're looking at melons to some extent. So just kind of more broadly, our experience with melon is on a couple of different fronts. A number of years ago, we looked at the homologous genes in melon to some of the transcription factors we identified in tomato. And in one case in particular, the RIN gene knocked out a very similar gene that was induced during ripening and had a pretty dramatic effect on the ripening. So very similar to what you saw in RIN. It slowed it way down. Didn't stop it completely, but certainly slowed it way down. So at that level, we know that there's some common control. Melon, as I mentioned before, is very interesting because you have what are referred to as climacteric and non-climacteric melons. So climacteric melons produce a lot of ethylene. They, if you inhibit that ethylene, you can control the softening, the maturation of the fruit. Interestingly, in melon, if you block ethylene synthesis or ethylene perception, and there's a number of ways to do that, the color change still occurs. So in a climacteric melon, like a cantaloupe, you can block ethylene, you still get an orange flesh, but now all the aroma that you really associate with melon is gone. The fruit are firmer. So you've blocked a lot of ripening, but you've not blocked that color change. Okay. So so let's set that up. in a non-climacteric melon, like a honeydew melon, you don't have that big production of ethylene, but you also have some other ethylene effects that appear to be impaired. So, for example, in cantaloupe harvesting, you harvest a cantaloupe when it slips. That is when the fruit abscisses off the vine. It literally slips off the vine. A honeydew doesn't slip. And that's an ethylene controlled process. That's the abscission of the fruit from the plant is controlled by ethylene. So it appears a honeydew has just a bigger issue with ethylene, maybe than just the ripening of the fruit, but they still ripen. So yeah, this is something that, and particularly what's going on with the carotenoids, how come they're not responding to that ripening signal? So that's what my student is particularly interested in. When people have done crosses with climacteric and non-climacteric melon, it's fairly straightforward to map just a couple of loci that seem to be largely responsible, indicating it's probably a couple of primary genes that are different. But some of what always concerns me about a crop like melon, and pepper is another good example, where you have within the species or within very closely related species accessions that do and don't need ethylene. It just makes you think that maybe you really are looking at mutants, right? That your non-climacteric is just an ethylene mutant of some type. So I think that's something very interesting to explore. It's not something we've done, and I know other groups are looking at that. So it's a good question because I think melon in particular is going to be a good system to get a better insight into ripening control and some of the nuances of ripening control.
We're almost at 1 o'clock, so we'll do one last question. You've explored flavor genes. What have you found are the most impactful gene families, transcription factors, or specific genes that impact flavor in fruit?
Okay, Ashlyn, very nice to hear from you. It's been a long time. So Ashlyn was a summer intern here a number of years ago.
Very great. Nice to have you here, Ashlyn.
Yeah, so they come back. That's great. So yeah, so what are the most impactful gene families? Boy, that's a loaded question. So certainly, you know, some of these regulators we look at will influence everything, and including flavor, right? So you can have very profound effects. You know, and what is flavor? That becomes an interesting question to begin with. So the industry will measure and is, you know, pretty accurate in saying flavor is a kind of the balance of sugars and organic acids, and they're both readily measurable. So you can get a good sense of flavor by just knowing your sugar and your acid content. But folks at the University of Florida that you know well, and others, have also shown that there's a lot of impact on flavor from volatiles. So understanding that chemistry of how certain volatiles are produced also helps us understand how flavor is achieved. And in fact, there's very nice work showing that in the context of fruits with the same sugars and acids, you can have very different perception of flavor from people in taste panels, all tracked back to how the volatiles differ, because those volatiles influence your perception of how you perceive flavor and even texture. You know, so when the right texture and the right juiciness can influence how you perceive flavor. So this is a lot of wind I'm blowing here to say there's a lot of genes that are important in flavor. It's a very complex trait, but some of the simple ones, if you want to know specifically, are going to come back to the genes controlling sugar, acids, and volatiles. Now, the transcription factors and some regulators that are way up in the hierarchy of control are influencing all of those things. So then you get very dramatic effects. But I think the keys to understanding flavor are really in the genes that are in the biochemical pathways that produce sugars, acids, and volatiles. And again, all of those are not single molecules. Those all represent a complex array of molecules, lots of different biochemical pathways leading to their relative profiles in the mature fruit. And then to complicate things even more, when it comes to flavor, there is no perfect flavor. What I think is a great tomato, the next person might think, well, that's too sweet or too acidic, or other flavor notes or attributes that they appreciate more or less. And you see that when you travel the world, you'll find in certain parts of the world much more acidic preference of the tomatoes that are being sold. Others, typical of kind of the US, we really like sweet things, and we tend to go to the sweeter varieties. But so flavor is a tough one. It's very complicated. And yes, there are many important genes. So that's job security. So that's a good thing.
That's a good thing. All right. Well, thank you again, Dr. Jim Giovannoni, for taking the time today to chat with us. And thank you everyone out there who joined us today for this virtual chat. And please join us for BTI's next Breaking Ground discussion series, Tuesday, October 18. We'll be joined by Dr. Murli Manohar, who will talk with us about his work to develop natural crop protection products that help farmers reduce disease and increase yield without harming the environment or human health. So Murli used to be here at BTI. He was in Dan Klessig's lab, and now he's with the company Scribe Bioscience, which was founded out of research done in Dan Klessig's and Frank Schroeder's lab. So that'll be really interesting. They're doing some really, really cool stuff. He's been zooming around, checking on field trials. So I think we're going to have a really neat one next month. We can hear some of the stuff that he's been up to. And again, that's October 18, noon, Eastern Daylight Time. Make sure we'll get that. It's Eastern Daylight Time. And you can read more about BTI's current research and many other neat stories about BTI science in our annual report, which you can find online at btiscience.org/annual report. And I'm going to paste a bunch of URLs in here that I'll be reading. So you can register for Murley's Breaking Ground at btiscience.org. There's a little thing right on the homepage. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. And thanks again to everybody out there for joining us today and for your interest in the Boyce Thompson Institute. And have a wonderful day and be well. Hope to see you next time.